I don't know if it's just me, but something seems to be up in the air these days. Is this a sign for a party? Imagine a flashing lights, a lot of people, and loud music playing. If you're the life of the party type, you might find yourself drawn to the crowd. But if you're like me, who is more on the values, peaceful, me time side, I'll step back for a bit once I find myself overwhelmed by the noise. These physical or chemical changes in our environment that can cause us and other organisms to respond are known as stimuli. The way we respond to the stimuli is referred to as taxis, particularly our movement toward or away from the stimulus. An organism exhibits positive taxis when it moves towards the stimulus and negative taxis when it moves away. Meanwhile, the ability to react to a stimulus is referred to as irritability. To further understand these concepts, some curious humans in the lab introduced me and my earthworm buddies to various stimuli, more specifically, touch, light, heat, moisture, and chemical stimuli were applied to our anterior, middle, and posterior parts. Even though it was a bit painful from our end, these five humans wanted to find out which part of our bodies is the most sensitive and observe where we'd wiggle off in response to the various stimuli.
Hold up. What's that smell? It smells like a whole setup over there. Oh, look. There's a dissecting pan, some tissue paper, a dissecting needle, an alcohol lamp, a card with a hole in the middle, a flashlight, and oh no, Silo? Gross. Why is this here? Oh, I think I know what's going on. I've heard the rumors from other worms. They say three dissecting pans are prepared before the experiments begin. One pan's lined with damp tissue paper where worms are stored. Another pan's also covered with damp tissue paper where experiments are conducted. And the third pan, half damp, half dry, sounds like a test to see if we will move toward water or away from it. Then comes the poking. Oh, the dissecting needle test. They poke our posterior, our anterior, then our middle. And that's why there's an alcohol lamp. The pan will be hovered above the flame, targeting our posterior, anterior, and middle. Next, I heard a flashlight is used in a dark room. Light shines through a card with a tiny hole aimed right at us. And finally, the worst part, a tissue soaked in xylol is brought near us.
Now, what do earthworms like? From these setups, what would they move towards and away from? When earthworms are placed in a pan with two halves, one moist and one dry, what do these little creatures like? We can see that, in response to moisture, the worms moved from the dry half of the pan to the moist half of the pan. This is hydrotaxis, or as the name suggests, response to moisture or hydration. Earthworms exhibit positive hydrotaxis, meaning that they move towards moisture. This is because earthworms are primarily made up of water. Actually, about 75% of their body weight is just water alone, so moist environments are preferred to prevent dehydration, according to the USDA Natural Resources Conservation Service 2009. Particularly, earthworms breathe through skin absorption. Thus, their skin must remain moist to allow for oxygen to effectively diffuse through the earthworm's skin. This is sensed by epidermal receptors on the lateral and ventral surface of the earthworms, allowing them to sense the differences in moisture between the two halves of the pan. This is also why the setups are always with moist tissue, so that these little creatures can move around and respond to the other stimuli. Response to touch or thigmotaxis. Now, if they sense abrupt touch, what do? We can see that through a representative worm, the earthworms twitch and move away from a sudden, sharp touch from the dissecting needle. For the anterior part of the earthworm moves its head back away from the stimulus. At the middle, it may be less noticeable, but similarly, it curves its body away from the source of the stimulus and eventually moves away from it. When touched on its posterior part or its end, we can see that it actively moves away from the stimulus by moving its entire body forward. Essentially, it attempts to escape the touch of the dissecting needle. They all observe negative thigmotaxis, meaning that they move away from the sudden, aversive touch. When they are suddenly touched or poked, they reflexively escape. However, earthworms also present with positive thigmotaxis to the surfaces they move along. Their natural tendency is to stay in close contact with these surfaces just as they burrow into the soil. They detect these through the epidermal receptors, which are sensitive to touch.
What if the stimulus was a bit more intense, like in the heat setup? They would move away from the source of the heat, right? Yes, because like many creatures, these earthworms do not like extreme heat. From the heat emitted by the alcohol lamp from below the pan, we can see that the earthworms actively move away from the heat stimulus through this representative earthworm. We can see that once they feel the heat, they move back and avoid it, and eventually redirect their movement. Then, when the middle of their body is exposed to heat, they try to escape the source of the heat. Their end also moves away from the source of heat. They all observe negative thermotaxis because earthworms are averse to high heat. This is because generally, these earthworms prefer colder temperatures and are actually hurt by very high heat, like the one emitted by the alcohol lamp. Similarly, the epidermal receptors respond to the thermal stimuli in this experiment, which are tactile and can also sense the presence of heat on the earthworm. And when exposed to light, we can also see that it moves away from this concentrated stimulus through this representative earthworm. When the head is shown with light, it moves away from the source of light. Through the middle, however, the entire worm seems to escape into the dark area. At the end of the worm, we can also see that it moves away from the source of light. They all exhibit negative phototaxis, where they move away from light to survive. This is because they prefer to actively seek out moist and dark environments underground, as these are the conditions needed for their survival. Dark environments with less light allow them to avoid drying out and be protected from predators. Though earthworms do not have eyes, they have single-celled photoreceptors that are found dorsally and are most numerous along the first segment of the earthworm, or the prostomium, followed by the peristomium, or the second segment. Each photoreceptor cell contains a nucleus, a network of nerve fibers or retinella, and a transparent L-shaped lens called a phaeosome. But how about chemicals that they encounter in the environment? How do they react to them? Through the experiment, we can observe that earthworms move away from the silent chemical that they sense. The earthworm keeps moving away and avoiding the chemical stimulus. The earthworm entirely crawls away and avoids the chemical stimulus. Or similar to the head, it keeps moving away from the chemical stimulus. They all observe negative chemotaxis from xylene, as this chemical is harmful and highly toxic to the earthworms, so they want to avoid it. Particularly, earthworms sense these chemicals through odor cues that are processed by their chemoreceptors on the anterior segment, or prostomium, and along the body's epidermis. More specifically, its buccal receptors respond to olfactory and chemical stimuli, and these can be found in the buccal epithelium.
Now that we've poked, heated, and shown some light on these earthworms, let's unearth what those responses truly mean beyond the lab. Healthy soil equals healthy food systems, and one way to tell is by observing the presence and activity of earthworms. Earthworms are bioindicators of soil health, meaning their presence or absence can tell us more about the overall condition of the soil environment. When they decrease in population or are less active, it often signals soil degradation, pollution, or loss of organic matter, all warning signs that point to an unhealthy ecosystem. Although relatively tiny organisms, earthworms are key to maintaining a balanced ecosystem. By burrowing and casting, they improve soil aeration and water drainage. They also facilitate nutrient cycling by decomposing organic matter and promoting ecosystem productivity. However, current research by Adomako et al. 2025 reports that earthworms respond to environmental stimuli, such as climate change, with elevated soil temperatures affecting their physiology and microbial associations. Thus, studying how they respond to their surroundings can inform the way we can conserve them and thus safeguard soil biodiversity and function. Finally, earthworms serve as valuable model organisms for studying animal behavior due to their simple yet well-organized nervous systems, which exhibit real-time observable behavioral responses. As illustrated in our experiment, they display innate behaviors such as turn alteration and tactile responses. Electrophysiological and imaging studies by Nakashima et al. 2011 revealed that earthworms have mechanisms for learning and memory, along with decision making. Overall, understanding earthworm responses reminds us that these small yet vital organisms can tell us more about our ecosystems and ourselves.